Now we've covered how natural selection and drift layer with migration, right? And the relationship and the push and pull between them. So now let's move on to kind of some real world applications of migration and thinking about migration in terms of conservation. And migration has costs and benefits, pros and cons in the context of migration. So we'll go through two examples of this that kind of illustrate these principles. All right, the case study we're going to focus on is domesticated versus wild rainbow trout. But this is really an example of a wider problem. We domesticate critters for a lot of reasons, and we help them by artificial selection adapt to our needs and to the environment that we create for them. And so those are local adaptations, all right? And so they have wild counterparts, right? So members of the same species who are wild. Most of our domesticated organisms, animals and plants, have them. And so what happens then when the domesticated organisms escape into the wild and start interbreeding with the wild population? And in this particular case, rainbow trout are endangered or threatened in most of their ranges. So they're already facing challenges. And now we're now we're inundating them with genes from a population that was adapted to a different type of environment, right? Domesticated rainbow trout have been bred to grow fast and to get big so that they provide more meat. And in this case, it's not a matter of escaping into the population. Humans actually intentionally release trout, domestic hatchery fish is one of the ways we say it, into the wild for sports fishermen, so for sport, or for the fisheries industry if we're talking about salmon. These things are happening, it's pretty common for this to happen, and for other species beyond rainbow trout. So they try to do things to keep the hatchery fish from breeding with the non-hatchery fish. Many of the fish that are hatchery fish, they attempt to sterilize before they release them, but not all attempted sterilization is successful, and so there is interbreeding happening with the wild rainbow trout. So we're creating gene flow between two different populations of trout that have been locally adapted to different conditions. So what are the consequences of this, especially for the wild trout that are already threatened, already endangered in some places? To understand this impact, researchers set out to do an experiment. So they identified several behaviors in trout that appear to have a genetic basis. And so those behaviors are foraging and then their anti-predator behavior. So in your notes, you don't have these pieces, so you might want to add them as we go. So foraging, trout feed at the surface of the water. So they're looking up at bugs and things like that that are falling on the water. But the challenge there is that predators are more likely to find them when they're at the surface. So when there's a predator around, the fish stay hidden near the bottom. So they're not coming up to feed, they're staying low and maybe in crevices under rocks and things like that, but they're staying near the bottom. So foraging behavior is feeding at the surface, and then anti-predator behavior is hiding at the bottom of the stream. So to look at the impact of gene flow from domestic fish into wild rainbow trout, they use the same genetic crossing that we talked about in Unit 2 to create fish with a mosaic, right, of the two parental types, both for phenotypes and for genes. And so they also created this kind of gradient of the percent of genes from the domesticated trout. So some fish had very little and some fish had almost 100%. And by doing that, they can look at the impact of those genes on behaviors. So they place the fish in an environment that replicates wild conditions. So they place them basically, this image, this lower image, is one of their experimental ponds. Okay? And they simulated, this is the best part, I think, they simulated predator attack by taking these plastic blue herons that they bought at like Home Depot and just dunking their heads in the water 3 times. And then they recorded what happened, what the fish did, what behaviors they showed. So in particular, they focused on how quickly the fishes started feeding at the surface again after the predator, which is kind of a measure of cautiousness, right? And then just their general activity level. So how often were they out from their hiding spots? How often were they away from the bottom of the pond? These are their findings, and I want you to notice that there are black and white dots on these graphs. And so the black and white dots are either prior to feeding, which means the fish were hungry, or it was an experiment done after they had just fed the fish, so they weren't hungry. All right, so you're going to have a question that pops up on the side asking you to interpret these graphs. So take a minute to look at them, and then we'll come back together. Good luck. So let's come back together and go over kind of the general conclusions we can draw. Your activity asked you about specifics, but what are we really seeing here? What we see that's going to impact conservation and survival of our wild and endangered rainbow trout is that these domestic fish, so domesticated fish, seem to have lost their anti-predator behaviors, so their predator avoidance behaviors. Predator avoidance behaviors. And this isn't learned. This is genetic. These are phenotypes. These are a behavioral phenotype. And so if they get these genes, they're also going to lose their predator avoidance behavior. But they aren't in a tank, right? That are being fed by humans and have nets over them to keep out things like herons. They're in the wild. So when domesticated trout breed with wild, they decrease the population's fitness by causing them to act in ways that attract the attention of predators. So in this case, when we're talking about conservation and migration, this is an example of migration leading to outbreeding depression. And not being a good thing. And you can see it in these graphs, right? So on the x-axis is the percent of domestic alleles that a fish has. So if they're at 100%, their activity level is way higher than if they're at 0% domestic alleles. And if your activities level is higher, that means you're away from the bottom, you're not hiding, you're exposed to predators. In addition, if you have more domestic alleles, you are, especially if you're hungry, right? And that's why they did those 2 separate experiments. All of this is exacerbated when they're hungry. If you have all the domestic alleles, you don't take very long to come back out after you've seen a predator, which means the predator's probably still in the area, whereas the wild trout are much more cautious. That's outbreeding depression. Our next example of migration and conservation and how they impact each other is way more complicated. This is the story of the Florida Panther, and you may have already heard some of this, if you grew up here especially. But basically, in the mid-90s, the Florida Panther was solidly in that extinction vortex that we talked about during genetic drift. Its census population was low, less than 30 individuals, and I believe its effective population size was like less than 8 individuals. And folks concerned about the conservation of the Florida Panther and researchers thought the genetic drift and inbreeding were overpowering natural selection and allowing all these detrimental alleles to build up. And they thought that because they were seeing all these deleterious effects in the panthers. So the population of panthers had increased heart defects. Some of them weren't even reproductively, couldn't reproduce. They were sterile because they had undescended testes. They had skeletal malformations, especially in the tail. They had these bent tails. Their sperm also had abnormality. So that's what this graph over here is illustrating. If you look at the sperm, percent of sperm with defects, and we look at cheetahs, tigers, leopards, domestic cats, we see all of them have very low amounts of sperm defects. But if you look at the Florida Panther, it's up at like 40%, right? So it seems like deleterious alleles had become fixed in the population. And the last challenge that was facing them was diseases. So if they have an effective population size of 8, they're essentially identical genetically. And so they don't have much of a reservoir to adapt to diseases. But researchers didn't know it was genetic drift that was the problem. So they had to test for it. And so they collected data on heterozygosity of the Florida Panthers. And so what I want you to do now is on the left side, you're going to have an activity pop up, but it's going to look exactly the same as this, except I can't insert these figures. So I'm going to ask you to just choose A, B, or C, which answer you think is correct. All right, but the figures will be on the slide, not in the question. All right, good luck. So if genetic drift was happening, we would expect to see very low heterozygosity for the Florida Panthers. Because of all these populations, they are the smallest. So it would be B. So hopefully you got that. That's kind of a bit of a review of the last unit. And if you didn't get it, or if you have any challenges with any of these activities or the content, please post to the discussion board and I can answer there, or we can set up an appointment to talk over Zoom. I definitely don't want you all to feel like I'm not there for you, even though we're not face-to-face anymore. I'm still here. All right, so moving on with this, here's the solution that scientists and then the managers came up with for the genetic drift problem, the lack of diversity genetically in the Florida Panther. They decided to try gene flow. So they brought in 8 panthers from Texas and released them in Florida to breed with the existing population of 30 panthers. All right, so another activity is going to open up, and I'm asking you to think about what are the potential costs and the potential benefits of this decision. And think of those based on your knowledge of what migration does. Think about what migration does layered with drift and what migration does layered with selection. Try to come up with 3. All right, let's come back together. You've now kind of made some predictions of what could happen. And so here is the actual data, what they saw. when looking at this first graph, this is just the population size, the estimated heterozygosity over time. They introduced the Texas Panthers right about here. Okay, and so another activity is going to pop up asking you what you would conclude from these slides or from these graphs. So I hope that was a little bit challenging, because the point is that it's complicated and that none of those answers were wrong. They were all right depending on what perspective you took on what happened. So let's go through this. Did gene flow work? Well, the population is recovering, both in terms of heterozygosity, right? That was the first graph, in terms of the fitness of individuals, at least for a year. And you didn't have this data, but also in terms of numbers of individuals. So the population was increasing. But think back to when we were talking about what it takes to balance genetic drift. What was that number? How many individuals a generation do you need to balance genetic drift? It's 1. And they released 8. This is where it gets complicated. 8 migrants was greater than the effective population size of the entire Florida Panther population. So we have, and I'm going to put it in quotes, we have Florida Panthers. But most of their genes at this point are coming from the Texas Panthers. So it's a legitimate question to ask, did we actually preserve the Florida Panther? Or did gene flow swamp that panther right out of existence? Right? And so what did we preserve? And again, this is why I think the success story depends on your perspective. We didn't preserve local adaptations. It would have been swamped by gene flow. Okay. We didn't preserve their unique evolutionary history. And what is a species other than its unique evolutionary history? So again, it would have been swamped. And swamped means overwhelmed. So I'll write that this time. Or here, I'll label this. Okay, so we didn't preserve their unique evolutionary history. We didn't preserve their local adaptations. What we did preserve was their ecological role on the landscape. We still have panthers playing the role of panthers in the ecosystem. And that's not nothing, right? That's important. Now, whether they're Florida Panthers or they're Texas Panthers, I think that debate is still raging. And honestly, when I look back about what wasn't preserved, I don't know how many local adaptations were left versus fixed deleterious adaptations. These managers and scientists had a really tough decision. They were working with a really challenging system, and they did the best that they could. And I want to leave you with this quote about the Florida Panther that really highlights this debate of what happened. Is the Florida Panther really recovering? There are definitely healthy panthers back in the Florida Everglades, but are they the Florida Panther? Or has that subspecies been driven to extinction by an invasive hybrid panther? Invasive, right, meaning migration and gene flow. The example here raises the critical question: is it better to keep some native Florida panther genes on the Florida landscape than none at all, which are the hybrid panthers, right? They have native Florida genes in them. Or is hybrid impurity worse than extinction for the Florida panther? Evolutionary genetics can provide the data, but not the answers to the moral dilemmas that these questions pose. When we think about conservation, when we think of the recovery of a very endangered species, something that's down to 30 individuals, migration starts to look pretty good. But it raises all kinds of questions about what is a species, and what is our role in preserving species, and what is the role of migration. All right, and so with that, we end the migration unit. I hope that these first two modules went okay for everyone. Again, please email me or leave notes in the discussion board if there were challenges. And the last slide in this slide set will just be, the questions that you should be able to answer by the end of. I will talk to you all next week.